Hello, this is the first part of the review of the 2018 Mid-Year MCQ. I'll now be going through questions 1 to 10. Please pause the video at any point or rewind if I'm not clear. Let's begin with question 1. Question 1, which statement best accounts for the difference in structure between amylose and cellulose? Okay, first and foremost, amylose is a form of starch. Cellulose, we also know cellulose can be found in plant cells. So what can account for the difference in structure? Well, the bonds are either glycosidic or peptide bond. This statement is not correct because in both cases, there's no peptide bond, only glycosidic bond. B, presence or absence of 180-degree rotation of alternate subunits. This is true. In the case of cellulose, there is 180-degree rotation. In the case of amylose, there isn't. C, presence or absence of branching in the macromolecule. In both amylose and cellulose, there is no branching, so this is not correct. Amylose, amylopectin, as well as glycogen, you can expect to see branching, but not amylose and cellulose. The subunits are either monosaccharides or amino acids. It is not amino acids in either case. Alright, both cases are glucose. Just as in amylose is alpha glucose, in cellulose is beta glucose. So this is also not correct. B is the best answer. Let's go to question 2. The diagram shows part of a polymer. This polymer is likely a polypeptide. How do we tell? Because you can see this peptide bond over here. So which molecule is involved in breaking the bond indicated by the arrow? When we perform hydrolysis, we require water, and therefore the answer is D. Even if you want to say that an enzyme is involved, this enzyme is not correct. This is amylase. Amylase is involved in the breaking of glycosidic bonds in starch. Now let's go to question 3. Question 3 asks you which is a valid deduction from these data. They show you purine and pyrimidine, and they show you the percentage of each present. They only gave you the identity of adenine, which is 28.2%. The other purine is guanine. Okay, and then the pyrimidines. Okay, so how do I know what is a pyrimidine? Okay, a student taught me this. Pyramids are sharp, right? And things that are sharp can cut. So C, U, and T are all pyrimidines. A and G are purines. So the answer here is B. Complementary base pairing is exhibited in base 1 as well as base 3. How do we tell? Right, so this is guanine. We see that it's 21.5. It's very similar to this 22.5. At least it is the most similar. So likely there's some form of complementary base pairing, and I suspect that this is C. This is likely T. Alright, we see that the percentage between these two are the most similar. Right, so likely there is some complementary base pairing between these two bases. So best answer is B. But let's look at the rest. Base 2 over here consists of 5 carbon sugar named deoxyribose. Actually, all of the bases would have been attached to a 5 carbon sugar. So not just base 2. C, the DNA content from cow contains more purine than pyrimidine. Okay, more purine than pyrimidine. Is this true? If you were to add up 28.2 and 21.5, actually it's very similar to 27.8 and plus 22.5. So this is not true, okay, because the percentage is almost the same. DNA is composed of 2 strands twisted around each other. While this statement is true, you cannot really tell this from the data. The data only gives you a percentage of the various bases present. So the best answer is still B. Next, let's go to question 4. Question 4 shows you the results of various food tests. They ask you which molecules are present in the extract. So breakfast test for disaccharide, positive. That means some sort of a disaccharide is present. Benedict's test, brick red precipitate. This disaccharide is a reducing sugar. Purple color means the protein is present. This means that glucose is not present. Clear solution means there's no lipids. Last but not least, blue-black color for iodine means that there's starch. Okay, so we look at the various options. Everything with starch is still an option. Everything with protein is still an option. Okay, but we see that there's no lipids present. So A and B is ruled out. We also see that the clinicistics test for glucose is negative, so C will be out. The best answer therefore is D. After all, this is maltose and it's a reducing sugar. Next, let's go to question 5. Shows you 4 different biomolecules. They ask and they say that one of them is responsible for the deposition of fatty plaques. So first and foremost, you need to find something that is a lipid. Okay, then which molecule is most likely to cause this unhealthy condition? A has to be ruled out because A is likely some sort of a polysaccharide. How do I tell? Each one of these units is a glucose unit. I can also see that there are glycosidic bonds, so A is out. D is also out because I can see peptide bonds, therefore it's some sort of a polypeptide. So we're only left with B and C. We look at B and C, actually both of them are fatty acids. I can tell because of this long hydrocarbon chain in both instances, and they also come with the acid part, fatty acid. Except one of them is saturated. This is saturated. And then this one below is unsaturated. How do I tell? The unsaturated fatty acid comes with a lot of double bonds. So which one is likely to cause fatty deposits? Well, you must understand that anything with double bonds means that they are kinks. If they are kinks, means that they cannot pack together as closely. So I give you an example. Here is a fatty acid, okay, that does not have any kinks. If they do not have any kinks, they can pack very closely to each other, almost like a solid state. That's why we tend to find saturated fatty acids come in a form of solids. Whereas unsaturated fatty acids, you will find that there are often kinks, okay, resulting in bends. And whenever there's a bend, it means that they cannot pack as closely together as they want to. Okay, so here's another bend, right? And then here, and then here's another bend. Notice that you cannot pack them as closely together, and as such, they tend to exist as liquid state or oils. Okay, so likely C is still okay. It's not going to be unhealthy. It won't pack as a solid. Whereas B is much more unhealthy because it can crystallize and solidify, pack very closely and form fatty plaques. Let's go to question 6. Question 6 shows you 3 statements. They ask you which of the statement is the reason for needing the pH to be maintained. Okay, first, statement 1 is not correct because they say that covalent bonds that maintain the enzyme structure will be disrupted in an acidic environment. It is not the covalent bonds that are disrupted by extreme pH. It is bonds that require differences in charge that will be affected by pH. For example, ionic bonds are affected by pH, as well as hydrogen bonds are affected by pH. These two are affected by presence of ions. 2, the enzyme only denatures at pH levels higher than the optimum. This is not true. Not just higher, it could be at the lower end also. So rather, enzymes only denature at extreme pHs. Right? So statement 2 is also wrong. Statement 3 is therefore the best answer, and therefore A is the best answer. That the three-dimensional structure of the enzyme is intact at the optimum pH. Outside of this optimum pH, less enzymes are in their optimum three-dimensional structure. Let's go to question 7. Which statement about the fluid mosaic model of the membrane structure is correct? Okay, this question relates to what we just said earlier, that when something is saturated, they can pack very closely to each other. Okay, something that's unsaturated, you'll find that they cannot pack as closely to each other because of all the kinks, the bends. So the more unsaturated the fatty acid tails or phospholipid, the more fluid the membrane. And this is true. So here we have something that's very saturated. The more unsaturated it is, the less likely the phospholipids will pack closely together in the membrane and there'll be more fluid. Temperature, okay, while the statements may be true that at high temperature membranes are less, actually at high temperature membranes are more fluid, low temperature they are less fluid. So these two statements are wrong for that reason. Let's go to 8. They say the enzyme lysozyme is secreted from tear glands and they can form deposits on contact lenses. They ask you which ingredient will be effective in removing this particular enzyme. We know that enzymes are protein in nature, so the only way to remove a protein is by using proteases to digest the protein. Okay, the rest aren't that effective in removing the protein. Technically, they still remain there. So for example, a pH buffer doesn't really do anything to the protein. Lysosomes may contain enzymes, but you typically find this in cells. You can't have them in an external environment. Last but not least, ethanol. Ethanol won't quite do anything to the protein, so proteases is still the best. Let's go to 9. 9 shows you 5 different cells. This 1 to 3 are plant cells, 4 and 5 are animal cells. And then they give you 2 different solutions, and then they ask you which one is likely to be placed in these 2 solutions. So if you look at 1 and 4, actually they are normal. Nothing quite happened to them. It is 2, 3 and 5 that has changes. If we look at 2, likely water entered the cell. 3, likely water left the cell. 5, water also left the cell. So in which solutions would you observe this happening? The best answer is C, because when you look at 3 and 5, water has left the cell. And this makes sense if it's placed in a sucrose solution. In a sucrose solution, there is low water potential compared to inside the cell where there's high water potential. Therefore, water would leave the cell. If you place it in plain water, which is in the case of 2, there will be high water potential in the plain water compared to inside the cell, and therefore water will enter. So the best answer would be C. Last but not least, let's go to question 10. They say the breakdown of hydrogen peroxide to water. Okay, so I'm going to write down the equation. H2O2 breakdown to water and oxygen is catalyzed by the enzyme catalase. Okay, then they want to investigate the effect of pH on the rate of reaction of catalase. And then they say they place potato cubes to hydrogen peroxide. What has potato cubes got to do with this reaction? Well, because you can find catalase in potato. Okay, so basically they are placing these potato cubes into hydrogen peroxide at different pHs. What is the dependent variable? The dependent variable is the variable that will change and is the one that you are recording. So one wouldn't be recording the mass of the potato because the mass of the potato shouldn't change. All it does is to contribute the enzyme, and the enzyme, as we know, is reusable. It will not disappear. The pH of the solution, this is not the dependent variable, it is more the independent variable. It is the one that we are changing. And we don't even change it at regular time intervals. We fix it for the entire duration of the experiment. Number of potato cubes, this is a variable you want to keep constant also. Okay, it therefore is the volume of oxygen that you want to measure at various time intervals. Okay, the more oxygen is produced, you know that the reaction is going faster. The less oxygen produced, the reaction is going slower. So D is the best answer. Okay, if you have any questions, please feel free to contact me. If not, you can move on to the next video where I'll go through questions 11 to 20.